Good afternoon, Jennifer. Welcome to our episode 8 of Making Reproductive Longevity a Reality. Hi, I'm Zhongwei here. I'm a clinician scientist in obstetrics and gynecology, and my main research interest is actually in unraveling the biology of ovarian follicular genesis in the hope to change the irrevocability of reproductive aging in women so that we can achieve reproductive longevity.
I'm Jennifer Garrison. I'm a faculty member at the Buck Institute in Novato, California, and I'm also the faculty director for the Global Consortium for Reproductive Longevity and Equality. And with the consortium, we are building a global ecosystem around female reproductive health. And we're doing that by fostering collaborations and dialogue between scientists and clinicians, industry partners and advocates. And we're doing that through funding, but also through things like this, where we're trying to go beyond traditional models. So the goal of the consortium is really to alter the societal balance toward equality for women by defining what leads to menopause and developing interventions that will slow or reverse it. And this truly is a global issue, and it crosses international borders. So this webinar series is designed to highlight some of the research that we funded. And today we are going to feature 2 talks from GCRLE grantees on very exciting topics related to female reproductive aging. Our first speaker is going to discuss how metabolic reprogramming might drive reproductive senescence. And our second speaker is going to tell us about how regulation of gene expression and maternal mRNA translation affects maternal aging. So we've talked in previous webinars about how the numbers of oocytes decline with age. There's also, a really important distinction there. There's also a decline in oocyte quality. That's a key feature of reproductive aging. So thinking about the first speaker, if the idea is that ovarian metabolic programming can drive reproductive aging, then that would imply that, targeting diet might be a viable strategy to consider. So Zhongwei, what do you tell your patients when they ask you about the efficacy of lifestyle modifications on their reproductive health?
That is something that I always emphasize, and I keep saying that the observational data are there, and we also know that the microenvironment in the ovary is so important. What goes in will ultimately have effect on your general health, and that includes the ovarian health itself. So I think it's very important that we try to understand what are the aging processes there's effect on the ovarian follicle, which is central for reproductive longevity. And I think thinking about metabolic processes, what can we eat in the diet, what can actually affect the broad big picture of how this affects a woman's general health, because the ovarian health is actually at stake. I think that really is what today's 2 speakers are going to share with us, isn't it, Jennifer?
Yeah, we've talked about this a little bit before, but the ovary is such a complex organ. It's got so many different cell types, so many different structures. And then on top of that, it's undergoing this complex dynamic remodeling through every cycle. So yeah, I think understanding how environment interacts with that process is really going to be really key to understanding what's happening. But egg development itself, right? The development of an oocyte from the beginning to fertilization is really complex and lengthy process. And one of the things that an egg does that's very unusual compared to every other cell in your body is that it builds up a lot of maternal messenger RNA, and that messenger RNA gets stored until the egg's ready to mature. And so that quiescent period can last a pretty long time sometimes. And when the egg is finally selected to be ovulated, then it has to very quickly, rapidly exit the sleeping state and grow. And that maturation step, fertilization, and then early embryonic growth really relies on that maternal mRNA to make the proteins that are necessary for survival. So yeah, I think our 2 speakers are talking about different processes, but they might have some commonalities between them. So I am delighted to introduce our first speaker. Her name is Zita Santos. Zita received her PhD at the Gulbenkian Institute of Sciences in Portugal in the laboratory of Dr. Monica Betancourt Dias, where she investigated molecular pathways which control the assembly of sperm tail during spermatid development, which ensures male fertility. So she started working on males, and then for her postdoc, she moved to the laboratory of Dr. Carlos Ribeiro at the Champalimaud Center for the Unknown in Portugal. Of course, we heard from Dr. Ribeiro in an earlier episode. As a postdoctoral researcher, Zita has been investigating the impact of metabolic reprogramming in the female germline on female fertility. and how this process influences female reproductive aging. So what she's discovered is that the process of egg formation, the female germline undergoes this shift in metabolism, specifically of carbohydrates, which is critical for female fertility, and also for sugar appetite. So, sugar cravings. She's now investigating whether female reproductive decline could be explained by an impairment of this phenomenon via metabolic reprogramming. Now, Zita is in Portugal, and it is the middle of the night in Portugal right now. So she recorded her talk for us, and we will ask you to please put your questions into the Q&A box, and we will discuss them live after Marco, our second speaker, speaks. But we will also move the discussion to gsarali.org offline in the next week, so that if you would like to interact with Zita and ask her questions about her talk, you can do that there. So welcome, Zita.
Hi, everyone. I would like to really start by thanking the organizers for the opportunity to be here today and tell you about how to bake sweet eggs using flies. So metabolism in very, very general terms is a process by which the nutrients are transformed into energy and building blocks by our cells so that they can achieve homeostasis. But actually, more recently, it has been demonstrated that metabolism is a bit more exciting than that. And it has a very active role in several cellular processes, such as chromatin biology and signaling. And it has been linked to a variety of different human diseases, such as cancer and aging. And so metabolism is fueled by what we eat and by the nutrients that we get from our diet. So the choices that we make regarding what we eat will have very strong impact on our health, lifespan, and also our reproduction. And so regarding reproduction, it has been shown there is a very tight link between nutrition and reproduction. And it's very important to have nutrients being provided at enough amounts and in a balanced way so that reproduction is achieved. And so both obesity and malnutrition have been implicated in infertility. And so also reproduction is a very complex process where a variety of different organs are in communication with each other via different hormones. And it's this concerted action of these different organs which leads to reproduction. And so the nutrients that we get from the diet, they will have different impact in these different organs, because cells from the brain, from the adipose tissue, from the ovary, they will have a very specific type of metabolism. And because of that, the nutrients will also impact them differently. And so fertility is actually an interaction between these different aspects: nutrition, tissue-specific metabolism, and physiology. And so the question is, how do these different nodes interact in order to ensure reproduction? And I think that a great organism to study this question is actually the Drosophila melanogaster. And why? So the Drosophila has a very powerful genetic toolkit, and also it allows for high throughput studies. Many genes are conserved between human and the fly, including the metabolic and the signaling pathways. It's very easy to manipulate all these different pathways due to these genetic toolkits that exist. And they have most conserved organ systems, including the reproductive system. And so in here, I'm just very quickly showing you how the reproductive system of the fly looks like, of the female fly looks like. It's composed of 2 ovaries, and each ovary has this chain of oocytes in different developmental stages. And it's very similar to the one of the mammalian ovaries. So it starts at the tip with a germline stem cell that divides and then gives rise to this chain of egg chambers, which are basically oocytes in different stages. And it's a very similar in terms of cellular organization and oocyte development to what happens during follicle development in the mammalian ovary. So we think this is a very powerful system to study this question and how to ask how these nodes interact to ensure reproduction. And so this is what I've been trying to do. And the way that I've been trying to approach is to approach each node at the time and ask smaller questions at a time. And so the first question that I asked was, what are the metabolic requirements of the germline per se? Okay. And so in here, I'm asking about nutrition. What are the nutrients that are required to supply to the organism so that it reaches successful reproduction? And so in the lab, we work with the Drosophila, and we have a very nice tool which allows us to manipulate each nutrient, which is a fully chemical diet. So it allows us to either add or remove specific nutrients. And so we decided to ask what was the role of dietary sugar in reproduction. So sugars are very important for providing both energy and a variety of macromolecules that eggs require for the oogenesis require for producing eggs. So we decided to start by asking whether this was a nutrient which was important for this process. And indeed, when we have flies fed on either a full diet or a diet without sugar, we see an immediate reduction of the ovaries of the flies which have been fed on a no sugar diet. And this then has a very strong impact in the production of the eggs, where we see a very significant decrease in the number of eggs that are produced by these flies. So yes, indeed, sugars from the diet are required for producing eggs, right? And so the sugars, they fuel these metabolic pathways in different tissues, right? As I mentioned before, and reproduction requires all these different tissues. But I really wanted to ask, what are the germline specific requirements, right? So does the germline have specific metabolic programs, and do these require sugars? Okay, so to ask this question, we decided to take a genetic approach by which we manipulated the availability of sugar directly in the germline and then ask whether this impacts reproduction. So the way that we did this was to knock down one enzyme, exokinase, which is important for the first step of glucose metabolism, and then ask whether this influences egg production. And indeed, you see a very strong decrease in the number of eggs produced by these females, which tells us that the sugar utilization directly by the germline is really required for reproduction. And we went further and asked which pathway of the metabolic of the carbohydrate metabolism is required for this. And there is this very important pathway, the pentose phosphate pathway, which is required for the production of lipids, of nucleotides. And when we knock down two enzymes of the pathway, we see exactly the same phenotype as we've seen when we knock down exokinase, with a very strong decrease in the number of eggs which are produced by the flies. So this tells us that this particular pathway is required for reproduction. So next we wanted to see where in the oogenesis process is this pathway active. And so the way that we decided to ask this question was to do an in situ hybridization, where we could see the mRNA of these enzymes being expressed. And so one of the things that we could immediately see is that there is an upregulation of this pathway at the stage where the oocyte is being defined. So the earlier stages of cell proliferation have very low levels of this pathway. So this tells us that the germline, through the process of oogenesis, go through metabolic reprogramming, metabolic reprogramming of the carbohydrates. And so far, I've shown you that there is a very strong connection between sugars in the diet and the production of the eggs, very likely by fueling this pentose phosphate pathway, specifically in the germline, and this is very important for reproduction. The question now is how? How does the animal ensure that there is a proper supply of sugars to feed the pentose phosphate pathway in the germline? So the way that we get the nutrients from the diet, it's by eating, right? So food intake is a process, is a behavior which is highly controlled. And it's a process that we can also very nicely study in the lab. So the way that we study this, the feeding behavior in the lab, is by using this behavioral setup, which allows us to see the fly feeding on 2 different sources of food. One, in this case, which has a sugar source, and the other one which has a yeast or a protein food source. And then we can see how many times the fly interacts, or we can sense that this setup can sense how many times the fly interacts with the foods. And this gives us a number of sips. So it's kind of like a number of bites that the fly takes into this food source. So to give you an idea of what the data looks like, when we do an experiment where we deprive the flies of sucrose, and then we see how many sips the fly takes from either the yeast food source, which is kind of the protein food source, or the sucrose, we see that deprivation of sucrose doesn't do much to the number of sips that the fly takes on the yeast. However, it gives rise to a very strong increase in the appetite for sugar. And this is because the animal can change these nutrient preferences to meet the nutritional needs, okay? And so our question was, how does the pentose phosphate pathway in the germline, so this metabolic program in the germline, impact sugar appetite? And so we took exactly the same approach, where we knocked down this exokinase from this pentose phosphate pathway, and then look at what happened to the appetite for sugar of these flies, okay? And so in here, I'm showing you flies which have been fed on a low sugar diet. So the number of sips on sugar is quite high. However, when we knock down these flies for hexokinase, there is a very strong decrease in this appetite for sugar. And the same is true for the knockdowns of these 2 other enzymes of the pathway, which show exactly the same type of phenotype, telling us that the activity of this metabolic pathway in the germline instructs the fly to eat sugar. And this is what actually is going to fuel the metabolic pathway in the germline to produce eggs, right? So there is a germline modulation of sugar appetite, which allows the fly to get the sugars it requires for producing eggs. Okay, so again, how does that work? So we have the germline, which has this specific metabolism, and we have a behavior, but in between there's a lot going on, right? So how does the germline signal to the brain that it has to eat or not from this sugar food source? So the fly has a fat body tissue, which is simultaneously a storage organ, but it's also endocrine organ and an immune organ. It's a little bit like the adipose tissue of mammals. And it also produces, similarly to the adipose tissue of mammals, it also produces a lot of cytokines, which then act on other tissues and organs to modulate their function. And so one of the peptides which is produced by the fat body is FIT. So and FIT had been shown to be a satiety kind of peptide. So we wondered whether FIT could be mediating this sugar appetite by the germline. And so the first thing that we did was to ask if by manipulating the germline we had differences in the transcription of FIT. And FIT again is produced and it's synthesized by the fat body, okay? And so we were very amazed by this result, because we saw that both when we completely ablate the germline or when we manipulate the pentose phosphate pathway by knocking down these different enzymes, we see that there is a very strong increase in the regulation, in the transcriptional regulation of these, in the mRNA levels of FIT. And so it's very clear that the fat body can sense the metabolic state of the germline and modulate FIT transcription, right? And so now what happens if FIT is upregulated? So we decided to take a genetic approach and to combine a FIT mutation with a manipulation of the germline so that we could see whether we could rescue the phenotype by lowering down the levels of FIT. So again, in here, I'm showing you the phenotype of sugar appetite in here in flies, which have been fed on a low sugar diet. So the appetite for sugar is quite high. Now, when we remove the germline, we see a phenotype which is very similar to the one where we remove the single enzymes of the pentose phosphate pathway, okay? So we see a very strong decrease in the appetite for sugar. But now if we combine this with a mutation for FIT, we have a rescue of this phenotype. So this tells us that the germline modulates the sugar appetite via a secreted peptide FIT from the fat body of the fly. So now we have more or less a picture of how this works, right? So the germline pentose phosphate pathway somehow, which we still don't know how, signals the fat body to then secrete this peptide that acts on the central nervous system. And so it's the interaction between sugars from the diet, fueling the metabolic pathway, the pentose phosphate pathway in the germline, which then sends a signal to the fat body and regulates the central nervous system, which then modulates both eating behavior and fertility. Now, the question now is what happens in aging, right? So what happens during a reproductive decay? From these different nodes, what are the changes that occur during aging, which could explain the decay in fertility in females? So in Drosophila, and this is what we aim at studying now with the funding from GCRLE. And so I just wanted to show you that Drosophila also undergoes this female reproductive decay. So we can see that as the fly ages, the number of eggs produced per female starts decreasing very drastically, and also the viability of the egg starts to decrease. So the percentage of unviable eggs start to increase. And so we want to understand how. What are the molecular mechanisms behind this? And from these different nodes, what are the most important components of these different nodes in this process of reproductive aging. So we will start by asking how tissue-specific metabolism changes, or if and how tissue-specific metabolism changes during aging. And this is what we hope to do within the next year. And so with this, I would like to thank very concretely the people in the Ribeiro Lab, which have been great, and especially Carlos Ribeiro, my supervisor, for allowing me to start this project, this very exciting project. All the facilities at Champalimaud, which help us with our work daily, and our funding agencies. Thank you so much for your attention.
I think that is an amazing and very exciting talk, especially understanding, sugar metabolism. And I think the mammalian oocyte, we know that it's quite unique. It needs its supporting cells to actually produce metabolites of sugar so that it could be used for subsequent development and for the growth as well during oogenesis as well as folliculogenesis. So it is my pleasure now to introduce Dr. Conti, who is the former director of the Center for Reproductive Sciences at the University of California at San Francisco. His research spanned 40 years of work on the physiology and molecular biology of cell signaling. And for several years, Dr. Conti's research has now been focused on the molecular mechanisms underlying the final stages of maturation of the female gamete. Today, his talk will focus on the mechanisms of gene expression in the oocyte, their role in oocyte developmental competence, and possible defects associated with oocyte aging.
Well, thank you. And first of all, I would like to thank Jennifer and you for inviting us and for giving us the opportunity to present our data. Let me share my screen. And at the outset, I need to acknowledge the people that have been doing the work that I am presenting today. So the project on aging and translation in the oocyte was started by Federica Franciosi, who has done some seminar observation on that. Federica is now back as a faculty at the University of Milan. I should also acknowledge two physician scientists, Hakan Kakma, Beisu, who spent a lot of time trying to relate finding in the mouse to the human oocyte. And finally, Enrico Maria Daldello and Susan Luong are responsible for all the molecular studies on RNA-seq and the interpretation of the data. But I should also present the portrait of a very important component in the lab, which is the mammalian oocyte. As you can see here, this is a fully grown mouse oocyte that we say is arrested at the germinal vesicle stage because of the presence of a large vesicle, which is the nucleus and nucleolus. The oocyte has reached this stage after a long developmental journey, which in a female actually starts during fetal life, and in women lasts up to several decades. In spite of this long journey, this fully grown oocyte is still immature and is not able to sustain fertilization and embryo development. To become a fertilizable egg, this oocyte has to undergo a process which is called Oxide Maturation, which lasts a relatively short time, 15 hours in the mouse and 24, 30 hours in a woman. And we can follow this process under the microscope. And the oocyte that re-enters maturation undergoes two meiotic division, first meiotic division and second meiotic division. It loses the germinal vesicle, assembles the spindle and the chromosome, and then in oocyte, the spindle does not stay in the middle of the cell, but moves to the surface of the oocyte itself. And this is required to allow a highly asymmetrical division where 50% of the chromosome remain in the oocyte proper and the other 50% are discarded in this small structure, which is called polar body. At this stage, the oocyte is now fully competent to be fertilized and to sustain embryo development, and it's often called an egg. If the egg is fertilized, the oocyte resumes the second meiotic division, release the second polar body, and then goes on into embryo development. So I have shown you before that these nuclear changes that we can record under the microscope, but there is another major change that occurred during this transition, and that is actually called cytoplasmic maturation, which has no real morphological correlates. But both nuclear and cytoplasmic maturation are essential for nuclear reprogramming and to produce a zygote that is now totipotent. So what is very remarkable about the oocyte is the fact that all this complex transition up to early embryo development occur in a special environment. Gene expression in these cells, unlike somatic cells where DNA synthesizes RNA and then protein is synthesized, in the oocyte there is no messenger RNA production, and gene expression is entirely dependent on regulated translation of messenger RNA that have accumulated in the oocyte during the prolonged growth phase. So our lab has spent quite a while trying to understand rules and regulation of this translational program. And we now have some tools that we have been using to study translation at the genome-wide level. And using this approach, we have now a blueprint of what's of the translation that is occurring during this short period of oocyte maturation. And we know that in mouse oocyte, about of the 7,000 messenger RNA that are present in the cell at this stage, about half of them continue to be constitutively translated during oocyte maturation. However, a quarter of the message present, and we know that they code mostly for protein that are required for growth, are progressively blocked in terms of translation and eventually degraded. At the same time, another quarter of messenger RNA now become activated in terms of translation. And this message whose translation is usually activated, we found the code for protein that are essential for the cell cycle, for transcription, and to organize the machinery that is required for epigenetic remodeling of the chromatin in the embryo. So therefore, we now know that there is a major switch that is going on during oocyte maturation with a major reshuffling of the translational program. So, and I will show the next two cartoons to try to highlight some of the feature of this regulated translation. So in this cartoon, I have put together the components of a complex which is called Polycon Complex, which is a complex that is required for repression of translation because it's involved in methylation of a residue of histone H3.3, which is essential for repression of translation. Now, if you look at the traces that I have around this complex, and these are traces derived from RNA-seq, where we monitor the rate of translation of the messenger RNA coding for the components of this complex, you see that all this translation increases dramatically during oocyte maturation, indicating that the assembly of this repressive complex occurred during oocyte maturation and eventually will have its role in the embryo, as it has been shown in several publications. Another complex that is thought to be very important is the Cohesin Complex. This is the glue that keeps together homologs during the first meiotic division. And again, you see that the translation of the different components of this complex is quite variable, indicating that perhaps this complex is a lot more dynamic than what is actually thought about. So over the years, we also have tried to identify situations where disrupted translation is associated with altered oocyte quality. And we have identified quite a few of them here, reported 3 of them. But in summary, therefore, we can really think that the translational program is essential to generate a good quality egg. So on the basis of these premises, we have decided to determine whether translation has anything to do with the most common form of decline of oocyte quality. And that is associated with fertility, aging, and decline in fertility. Here you see, it's well known that in females, aging, fertility decreases with aging. But what is more important are these data that I have replotted from the CDC, where it's reported the rate of live birth after donor transfer or non-donor egg transfer. And as you can see, women that are progressively aging become less efficient, and we have a major decline in live birth in this IVF setting. However, if these older women receive donor oocyte for young women, the rate of live birth is basically normal. And this really underscores that infertility that is associated with aging is entirely dependent on the quality or number of oocyte produced. So to determine whether aging may affect the translation, we have again turned to our mouse model. And what we again have used this genome-wide approach where we have compared translation of messenger RNA in young and old oocytes. And these are the preliminary data that we have, where we are plotting the ratio between old and young translation for each messenger RNA. And you see most of the messages are translated at this identical rate or similar rate in young and old oocyte. However, we can detect a subset of transcripts which are poorly translated in the old oocyte, as well a group of transcripts which is excessively translated in old oocyte, opening the possibility that indeed a disruption of the translational program is associated with aging. So in the lab, we have been studying for quite a while the regulation of translation of the messenger RNA that codes for interleukin-7. Interleukin-7 is a chemokine, cytokine that is very important for immune system homeostasis. And what we find that this cytokine is also produced by the oocyte. And what is more important, that we find that translation of the messenger RNA as well accumulation of the protein increases during oocyte maturation. But what is even more important, this regulation of translation and accumulation of the protein is sensitive to the environment in which the oocyte mature. And we have a major increase in translation and protein accumulation if the oocyte are maintained within the cumulus cells. And we find also that something similar is occurring in women, and we've done so by measuring the interleukin-7 level in the follicular fluid of dominant follicle of patients undergoing IVF. And what we find that follicle then yield a fully mature M2 oocyte has higher level of interleukin-7 accumulation than a follicle where maturation has failed and the oocyte are remained in the GV stage. Also, retrospectively, we know that if we compare interleukin-7 level in follicles that yield an oocyte that can be fertilized, the interleukin-7 concentration is higher than in follicles that are not produce an oocyte that is not fertilized or is abnormally fertilized. So we have been thinking of using the interleukin-7 both translation and secretion as a sort of canary in a coal mine to try to understand whether translation is also disrupted during aging. So we have done this following this strategy where we have taken the regulatory elements in the messenger RNA of interleukin-7 and fused them to a fluorescent reporter. And we have injected this reporter together with a control reporter for the injection in oocyte and follow the accumulation of the yellow reporter during maturation. And using time-lapse microscopy, what we find is that if an oocyte is maintained in prophase, in GV, there is very little accumulation of this yellow reporter. However, if we allow the oocyte to re-enter the cell cycle, we have a major increase in accumulation of the reporter. And by interpolating this data, we can actually measure the actual rate of translation of this reporter messenger RNA, and we find a major increase. Now we can, using this system, we can compare young and old oocyte, and what we find that in old oocyte, the translation of this messenger RNA is considerably decreased. We have repeated this experiment now with 3 different cohorts in aging mice, and we continue to see exactly the same phenomenon with a significant decline in the rate of translation of this reporter, suggesting that perhaps the translation of this reporter is faulty with aging. And we actually have a hint that something similar may be happening also in women, because if we stratify the interleukin-7 concentration data that we have by grouping the according to the age of the female undergoing IVF, we do find a significant decrease in interleukin-7 accumulation during aging. So on the basis of these premises, and with the support of the Global Consortium and the Buck Institute, we hope we will be able to consolidate this initial observation and confirm that indeed the translational program is affected during aging, and that may be, at least in part, the cause in the decline of oocyte quality. And if we are able to do that, we will also be able to detect these changes by monitoring, for instance, the translation of the synthesis of any given protein, and perhaps to correct this by modifying what's happening. So with this, I will stop, and I would like to thank again the support of the Global Consortium and the Buck Institute, as well as NIH for the support of these studies. Thank you.
Thank you, Marco. That was wonderful. Really exciting. I can't wait to see the results of that study. So we have questions in the question and answer box, but if you have any other questions for Zita or for Dr. Conti, please put them into the Q&A and we will ask them. And so I'm going to ask the first question. This is coming from Pavla Brakova to Marco. Are there any common regulatory elements in the transcripts that are over-translated versus under-translated during aging?
We have been looking at that. So we have published a paper last year where we show that a master regulator of translation during these stages, and actually that's work done by somebody in Spain mostly, is cytoplasmic polyadenation element binding protein 1. And we know that that's a master regulator that regulates both repression of translation and activation of translation during oocyte maturation. Interleukin-7 messenger RNA does have CPB elements, and we have very preliminary data that the levels of CPB1 protein may be altered during aging. But by mining other data that are available, we do see sort of footprints of altered expression of some RNA binding protein that may be important in the regulation of translation. And that's obviously something that we intend to pursue.
That's very exciting. I have a question, thinking about, how complex the ovarian compartment is and thinking about interactions between oocytes and other tissues and somatic tissue inside the ovary. Do the defects that you're seeing in translation with aging, are they originating autonomously in the oocyte, or could it be the results of some faulty interaction between the somatic cells in the follicle?
Actually, we are quite open to the possibility that both things are happening. So we have published papers showing that this program of translation in the oocyte is not entirely cell autonomous, and it's actually sensitive to cues from the surrounding somatic compartment. And I've shown you the data for interleukin-7, where translation of interleukin-7 messenger RNA in the oocyte is sensitive to the surrounding cumulus cells. And if you activate the cumulus cells, you have a further increase in the translation of this protein. And we think that this is very important. So we are quite open to the possibility that there are both autonomous defects in the oocyte, as well as failing interaction between somatic cells and the oocyte itself.
Right.
Yeah.
So Marco, I think that is very exciting, because my actually research interest is all about the microenvironment. And I see that you have actually gotten quite a number of samples from women who is undergoing IVF, the follicular fluid, and this somatic cells around the oocyte. They have shown this differential expression of the mRNA for IL-7. Can you tell us, is it really the aging process itself, or do you think it's linked to the fertility treatment itself? Because there's a lot of studies have shown that, maybe we're given such high dosages of recombinant FSH that's also causing this change. Do you think this is also aggravated by the treatment itself, or is it just an age effect?
Well, translational messenger RNA is a very complex process, which is sensitive to about everything. Obviously, energy levels, mitochondrial function. Obviously, if we mine our data, we do show that, for instance, translation of some important protein that are required for removal of oxygen radical is regulated during oocyte maturation. And it's also known that translation is sensitive to the energy and the present and metabolism present in the oocyte. And it's well known, for instance, that the mTOR pathway is a major regulator of translation. And we do have some evidence that we have published that activation of the PI3 kinase pathway, which is proximal to AKT and mTOR, may be a means by which somatic cells regulate translation in the oocyte. So it is quite possible there is complete integration between energy, metabolism, and translation.
That's interesting, isn't it, Jennifer? Because we have someone here asking about type 2 diabetes, actually, on the sugar levels. And do you think type 2 diabetes, this is actually probably for Zita, but I think Marco, you probably could answer as well. Do you think type 2 diabetes could have an impact also on female reproductive longevity?
Absolutely. I think, this is not our work, but is the work of a group in St. Louis was quite shown that the oocyte quality in diabetes model is obviously declined. So I think that's quite accepted.
Yeah, I think there's a really well-established link between ovarian function or dysfunction and metabolism. Certainly long before menopause, women with PCOS are prone to metabolic diseases like diabetes. And we know that from one of our earlier webinars that fecal transplant from PCOS patients can disrupt ovarian function. There's a lot of, I would say, indirect links too. But certainly with PCOS and diabetes, it's a bidirectional, it's not just in one direction. The higher levels of insulin produce more androgens, which then potentiates the PCOS phenotype. And so it's back and forth kind of dysfunction.
Yeah, indeed. So in polycystic ovarian syndrome, what we see actually, there are so much follicles, there's so much follicles growing, but yet they are not being ovulated. I'm just wondering if that sugar levels really is the one, because I've always been telling women that look after that, please reduce your sugar levels and try not to have so much chocolate and pizza and whatnot, so that your ovaries probably will react to all the treatment and whatnot. I'm not sure if Marco has seen that in your data as well. Anybody who are PCOS in your group that actually show some differences or outliers when you analyze the data, because I know from my IVF cohort that we look after, some of the women, they have so many oocytes, when we do IVF for them. But whether are they of the right quality and whether does aging actually, worsen the quality of these oocytes because of the microenvironment, sugar rich or insulin, pathways metabolic pathways that's being affected. Do you think this actually has any effect on the aging process as well?
Well, I'm not sure about that. Obviously, polycystic ovarian syndrome does have an effect on oocyte quality, and that has been published. I don't really I think I should refer again to studies. I wish I remember the name of the PI on those studies, where they're really showing that altered metabolism has a major effect on oocyte quality and ability to mature and to sustain embryo development.
Right. Do you think it will be affect primarily at the protein translation level, at mRNA translation?
No, not necessarily. I think it's a very complex of effects that affect both the oocyte itself as well as the microenvironment in which they develop.
I see. Right. Jennifer, there's this very interesting question. I'm not sure if you could answer for Zita, because they say that, someone was asking in fly, lacking a liver, proteins and fat may not be converted into sugar. But in human, we have a huge liver. There's undergo gluconeogenesis from fat and protein, making the sugar source very diversified. How do you address this shortcoming in the fly? Is it a good model to study this phenomenon? I'm interested too to find out.
Yeah, absolutely. As you know, in my lab, we work on both worms and mice, and I have in the past dabbled in flies. It turns out that Drosophila actually has very similar liver-like functions that are controlled by two tissues, the fat body and the enocytes. And enocytes have really a really striking functional similarity to mammalian hepatocytes, actually. So there are lots of genes and pathways that are conserved between the mammalian liver and the fly enocytes. And there's similar upregulation, actually, of many of the same pathways with age. So, for example, there's a lot of inflammatory pathways that are upregulated in the liver, in the mammalian liver with age, that are also similarly upregulated in the fly enocytes with age. And they also express genes for things like ketogenesis and for all of these metabolic processes that the questioner was asking. So, believe it or not, flies do have some liver type function.
Well, that is quite interesting, isn't it? Because even a fly who produces so many little flies from their egg laying, they still undergo reproductive decay. And it seems that something is ongoing that we need to study this further. There are two more questions for Conti, and I think these are very interesting questions. Then I ask another question. Will Tightness rejuvenation be able to restore ovarian function by recovering IL-7 decline over age?
Well, at this stage, I can't really talk about that. I mean, we don't know. That certainly is sort of a telltale of what's happening. I don't know if by itself it will be able to cause rejuvenation. I think this secretory product of the oocyte is fascinating, because one thing that we find, and data in humans have confirmed this, is that when you get the LH surge, you activate translation and secretion of interleukin-7 in the oocyte. At the same time, what the gonadotropin does, it upregulates tremendously the interleukin-7 receptor in the granulosa cells. And actually, I've seen in some genome-wide data in humans is one of the most upregulated genes when the ovary is stimulated, the follicle is stimulated by LH. So what this clearly shows that is something that is happening at this stage, that the oocyte increases secretion of this protein, and at the time of ovulation, the gonadotropin activate a system that is able to receive this signal from the oocytes. So we don't exactly know what this signal is important for, but we think it is quite important. And it is possible that may contribute to decline in fertility if we have a decrease in interleukin-7. But obviously, we cannot say whether this is the most important factor, and therefore we can't say that if we increase the level of interleukin-7 in the ovary and the follicle, we can restore oocyte quality.
That would be an exciting experiment to try. One last quick question from Pavla Brakova again. Do you have a sense whether the defects are in the recruitment of the transcripts to the ribosomes, Or is it more about the efficiency of translation? You know me, I'm very excited about ribosome profiling.
We don't know. I mean, it might be both. So I should point out that the reporters experiment that I have shown you uses the 3' UTR, or untranslated regional interleukin 7, but we have not fused also the 5' UTR. So we don't know whether regulation of the cap and recruitment of the small subunit of the ribosome is also affected. But there are other data that actually suggest that. And there is my former postdoc, actually, Andrei Suser, has shown that activation of mTOR may activate an activation of translation through recruitment of the cap. So it's another component, and we have not explored that. So it might be that is dependent of each individual messenger RNA, where you might have 5' and 3' regulation, and in some other cases, mostly 3' regulations. So that's an open question. And
yeah, I think understanding that specificity for individual mRNAs is going to be really, really important. Thank you so much. That was really exciting. I really am looking forward to seeing the results of this work. So please join us next week. We have another 2 talks from our JCRLE grantees. So on April 22nd in Singapore, on April 23rd in the Pacific Northwest, Mary Zielinski from Oregon Health Science University and the Oregon National Primate Research Center will tell us about her work. And Farners Emergant Riera from Northwestern University will tell us about her work. And these are talks that you do not want to miss. I also wanted to let everyone know that our final episodes, so we're going to have episode number 10 on April 29th or April 30th, depending on where you are in the world. We're going to have a panel discussion with thought leaders, Nicole Shanahan, who is the president of the BIA-ECHO Foundation, Professor Yap-Seng Chong, who is, of course, the dean of the NUS Yang Lin School of Medicine and the executive director of A-STAR in Singapore, and Dr. Jun Yoon, who's the president and managing partner of Palo Alto Investors, who's really an advocate for reframing women's health around this question. And so they're going to talk about the challenges and opportunities that we face in building this ecosystem, how to facilitate and accelerate translating basic discoveries into useful products that are really going to impact women's lives. And I think it'll be a really important and exciting discussion, and we will be taking questions. So if you would like to continue the conversation from today with Marco or Zita, we will transfer these questions to jcrle.org. So please keep asking them and go to jcrle.org to join the conversation. Zhongwei?
Yes, and thank you everyone for being a part of this journey with us. And until next week, please all tune again to Making Reproductive Longevity a Reality. Thank you very much.